In examining your blood film, you have these thousands of thrombocytes being seen under your microscope. However, click on the clinical details and you find that the patient is bleeding. How do you relate these thousands of platelets to a bleeding episode? And the most interesting aspect is that after many years, for instance, about 10 to 15 years, you go into the clinical details again, and there is new diagnosis for this patient who was previously essential thrombocythemia and now having myelofibrosis. How does this transformation occur between these two hematological malignancies? Very interesting. But just like we have always been doing, we always establish the basics, and it is always about proteins. What we are going to discuss today is about a protein that is called a calreticulin. This is a chaperone. When we say chaperone in science, we mean that it is a protein that is specialized in its function, causing the activation or initiation of other processes. And just for you to also get a clue, when we say a rogue chaperone, it is a mutated form of that specialized protein, meaning it's not going to produce or it's not going to deliver its actual function. Now, when we say calreticulin, calcium, reticulin, endoplasmic reticulum. So this is a chaperone that is found within the endoplasmic reticulum. And just as it mentions calcium, it is responsible for calcium homeostasis. Its structure is endowed to bind to a lot of calcium, thereby regulating how available calcium would be. And this is why it's quite related to cardiac functions. But back to our discussion. And even as it regulates calcium, its other function includes protein folding. Glycoproteins need folding after they are produced. Don't forget that the endoplasmic reticulum, the rough one, has a ribosomal compartment where protein synthesis occurs. So this calreticulin is a chaperone for folding of these glycoproteins. One other function of the calreticulin is gene expression. Calreticulin has affinity to binding to the DNA domain of some steroid receptors. These include the vitamin D receptors, the retinoic acid receptors, and the glucocorticoid receptors. In binding to their domain, meaning it is covering their response element within the nucleus. This resists the binding of these actual receptors, and therefore they are down-regulated. On the contrary, while down-regulating these steroid receptors, the calreticulin promotes cardiomyogenesis by activating GATA4, which is a transcription factor for cardiomyogenesis, as well as regulating calcium 2+. Also, one last function we will talk about calreticulin is its ability to activate the major histocompatibility complex class 1, recruiting it into protein folding, that is, loading of a foreign protein for cytotoxic T cell function, which is generally involved in immune surveillance of CD8. Having understood the normal function of this calreticulin, which is a chaperone, let's see, when we say a rogue chaperone, it's a mutated form of a protein which has lost its normal function and now causing the opposite of what it's meant to do. Mutant is a mutated form of calreticulin, and this has an affinity for MPL receptor. MPL receptor, or CD110, is a receptor for thrombopoietin. It binds to these hormones, ligands, or cytokines from conformational changes, sending signals and activating adjacent genus kinase 2, which is the JAK2 proteins, which are found on opposite sides within the cell. And when JAK2 proteins are activated, there is a signal transaction and activation of transcription that is sent directly into the domain or nuclear environment, causing the promoter region to begin gene expression, transcribing the portion of gene that is needed. When a ligand binds to its receptor, we expect either a proliferation, a differentiation, a cell migration, or apoptosis. In explaining this ligand-mediated receptor activation, and when we say ligand in this circumstance, we talk about the hormones like erythropoietin, thrombopoietin, and as well other cytokines, interferons, and growth factors. There is an interaction between thrombopoietin, which is the ligand in question, with its MPL receptor, which is the thrombopoietin receptor, CD110. And in this case, there is a dimerization between the ligand and the receptor signal sent downstream into the cell, where there are two proximal adjacent genus kinase 2, which is JAK2, Of course, there will be a transphosphorylation between these two adjacent JAK2 and a signal sent down to the signal transduction and activator of transcription, which is either STAT3 and a STAT5. There we say it is a heterodimerization between them, or a homodimerization which exists between a STAT3 and another STAT3. At the C terminus of this STAT, genus kinase 2 activates it. This activation sends a final signal into the nucleus where the promoter region is transcribed due to its activation, and the gene of interest will be produced in the end, and there will be proliferation of that particular cell that we intend to produce. This is the normal process by which the ligand activates a receptor to transcribe a particular gene. An interesting thing here is that the mutant gene has an affinity to bind to this receptor and causing this signal transaction downstream to the nucleus, even independent of its specific ligand. So this is a ligand-independent activation and signal transduction. In the course of this process, when the signal transduction reaches the nucleus, it continuously produces cells for the receptor whose ligand is being mimicked, and in this case, the CAR mutant mimics thrombopoietin. When the signal gets to the nuclear environment, it transcribes megakaryocytes, and in this case, CAR mutant causes a high number of megakaryocyte production. And don't forget that each megakaryocyte cell produces about 1,000 to 5,000 of platelets, and a large quantum of platelets is produced as a result of this mutant protein. Then the consequences is really relevant for this discussion. A high number of platelets is being produced, but these platelets are functionally impaired, meaning they are unable to cause aggregation and they are unable to seal an injury. This makes people also prone to bleeding despite the fact that there is a high level of platelets. And in addition, contributing to the cause of bleeding or being prone to bruises, high platelet level activates the ultra-large von Willebrand factor. And of course, the von Willebrand factor stabilizes factor 8. So when these ultra-large von Willebrand factors are activated, this means that they are going to be consumed as if there is an injury, lowering the level of von Willebrand factors and also causing factor 8 instability. This contributes to the bleeding episodes experienced by people who are suffering from essential thrombocythemia. In addition to the other symptoms that will be experienced, because of the high quantum of platelets that will be produced, the blood is thickened, leading into stasis. When blood stays at a point for very long, it means that the contact factors will be activated, having access or coming in contact with the vascular endothelium. When these clotting factors are activated due to stasis, they form a clot. This makes patients who suffers from essential thrombocythemia also prone to thrombosis. And when we talk about thrombosis, it could be deep vein thrombosis or pulmonary embolism. Regarding how this hematological malignancy ET leads into other malignancies such as the myelofibrosis. The high quantum of megakaryocytes produced within the marrow, which is in the hematopoietic stem cell, activates fibroblast production because there is high level of cytokines produced by the megakaryocytes. Therefore, fibroblast replaces the spongy flexible tissue within the bone marrow by a fibrotic tissue, which is scar tissue. These scar tissue are not functional compared to the stem cells within the bone marrow. They are unable to produce the right level of cells for the normal function of the blood system. And this is what we call the secondary myelofibrosis, because of the replacement of the spongy tissue with fibrotic tissue. Then your blood film, which was previously about 10 years ago, full of high level of platelets and probably seeing megakaryocytes within, because of the myelofibrosis, there is now abnormal cells and the bone marrow is forced to push out any level of cells that's available. And of course, immature cells are pushed into the bloodstream to solicit for the low level. And it pictures myeloid precursors like promyelocytes, myelocyte, metamyelocyte, and sometimes some blasts in there. You will see nucleated red blood cells as well. And this appearance is called leukoerythroblastosis. So this is the leukoerythroblastic picture that you would see, coming up with night sweat, fever, prone to infections, body weakness, and a lot of that. And just to finish with this, because the myelo is now fibrotic, meaning it is unable, hematopoiesis shifts from this primary origin of blood production into a secondary form, which is the medullary hematopoiesis, and this occurs within the spleen and the liver. And look at a strong grown body whose blood production is being shifted from the myelo onto these external organs. There is too much pressure on your liver, which was previously performing a metabolic activity, and therefore it begins to enlarge, and this is what we call hepatomegaly. And your spleen as well also enlarges, and this is splenomegaly, and the two combined we call the hepatosplenomegaly as a final appearance due to myelofibrosis. If you want treatment guidelines, there are a lot of treatment guidelines within Cancer Research UK, and I would advise that upon seeing any symptoms of this kind, you speak to your clinician, attend to an appointment by a hematologist, and they have very good treatment guidelines to assist you in your circumstance. Thank you very much for your time, and I really appreciate it. You click on the like button and you subscribe to this channel if you are interested in more educational videos of this sort. Goodbye.